So this is an animation by Gary Kaiser at the Community College of Baltimore County, Maryland, and I like to use it when I describe peptidoglycan synthesis because I feel like it's a lot clearer than some of the other videos out there. So let's go ahead and get started. The first thing that you need to know is that there's a group of proteins that are involved in peptidoglycan synthesis, and they are called autolysins. And what autolysins basically do is they break down peptidoglycan so that it can be built up again, refreshed, renewed. This also allows cells to grow. If you think about peptidoglycan cell wall as being like a cardboard box, if a cell wants to grow, it's got to break down that cardboard box in order to make it bigger. So that's what's going on here. So what Gary has drawn here is a peptidoglycan backbone. You can see the NAG and the NAM molecules alternating in blue and red. He's connected the NAM molecules using these green spheres. Those are the amino acids. And then the pink line that's between the green spheres, those are the crosslinks that join the NAM molecules in the backbone of peptidoglycan and make the cell wall stronger. So he's representing the autolysins using these purple and green spheres with an A in them. There are two types. You have the autolysins that break the crosslink between the NAM molecules, and you also have autolysins that break the glycosidic linkage between the NAG and the NAM. So you can see these guys getting busy here, breaking down the peptidoglycan so that a cell can grow. So those are the autolysins. After this happens, the next thing is that we've got to patch up the peptidoglycan to restore the cell wall. So this is done by a molecule called bactoprenol. Bactoprenol is represented by a peach oval that's inserted into the plasma membrane of the cell. Okay. So what's going to happen is that a protein is going to bring precursors to NAG and NAM to the bactoprenol, and it's going to actually attach them. So here we're attaching the amino acids to the NAM, then it gets attached to the bactoprenol. Next, the NAG gets attached to the NAM, and then bactoprenol is actually a lipid that transports both of those sugars across the plasma membrane and out to this area of the cell called the periplasm. The periplasm is where we're going to find the peptidoglycan cell wall. So now we have an open cell wall. Remember, it was broken down by the autolysins. We've inserted our patch of NAG and NAM, but the patch is not actually attached to the cell wall yet. So that's the activity of another enzyme. Transglycosylase. Transglycosylase is an enzyme that's going to come in and it's going to restore those glycosidic linkages. Trans means across and glyco means sugar, right? So we're going to add bonds between the sugars in our peptidoglycan patch, joining that patch to the peptidoglycan cell wall that already exists. So here comes transglycosylase. You can see it's making a bond between the NAG and the NAM, which somewhat affixes the patch to the rest of the cell wall. Next is another enzyme called transpeptidase. Trans again means across, and peptidase, peptid means like peptide, right? So that means we're going to join those amino acids from our NAM molecules together in order to form a complete backbone and to strengthen the cell wall. So here comes transpeptidase. You can see it's cutting off an amino acid and making a bond between our NAM molecules. So now our peptidoglycan is complete. Let's look at the whole process all together. So again, first thing that's going to happen is our autolysins are going to come in and they're going to break down the cell wall. This is what normally happens periodically. Again, this allows the cell wall to gradually be replaced and refreshed. It also allows the bacterial cell to grow. So here come the autolysins. They cut the glycosidic linkages between an old part of the cell wall. They also break the cross-link between the amino acids on the NAMs. This opens up the cell wall so the cell can grow. Next, our precursors to NAG and NAM get attached to bactoprenol. Remember, bactoprenol is that lipid. It's going to transport our NAG and NAM to the periplasm, the space outside of the plasma membrane where the cell wall is located. It's like placing a patch over a hole in your jeans. The next thing that's going to happen is transglycosylase is going to come in, and it's going to make bonds between the NAG and NAM sugars, kind of tacking that peptidoglycan patch into position. But it isn't until transpeptidase comes in to make the crosslinks between the NAM molecules that the peptidoglycan wall becomes complete.